Okay, this is really a great honor and pleasure to welcome Joan Roughgarden today. Um, we had the pleasure in the past to have Joan Roughgarden for some of our virtual conferences on the Holobiont, and as you will see in a minute, it is related to what we will hear today. So, Joan Roughgarden is Professor Emerita in the Department of Biology at Stanford University, and she is or was an adjunct researcher at the Hawaii Institute of Marine Biology at the University of Hawaii in the US. Um, Joan Roughgarden is very well known for her work in evolutionary biology and ecology, and her work is quite unique in bridging biology, behavior, and social theory. She has written very important books, including in 2004, Evolution's Rainbow, and in 2009, The Genial Gene. Both attracted a lot of comments, enthusiastic comments, also some interesting critiques. Of course, this is part of science, and these two books have been extremely important and influential. She has played a key role in debates about sex, gender, and sexuality at the interface between biology and society. And she has written more than 200 scientific papers on the Feed in Biomed website. We, I think, gave maybe 15 examples of those very impactful papers, but in fact, there are many more. Recently, Joan Roughgarden worked on microbiology, seeing among other things that microbiology is often quite, let's say, experimental in its practices. There is also a theoretical tradition in microbiology, but it has remained relatively small. And one thing that she started to do was to bring some theoretical insights to certain questions that have been extremely important in microbiology, either for a long time, for example, about lysogeny and bacteriophages, or maybe in some cases more recent work, typically on the microbiome and holobionts and hologenomes. And she brought very interesting theoretical perspectives about these questions. We shared with the participants of this seminar two papers, one which is entitled Evolutionary Optimal Phage Life History Traits, and the other published 3 years ago about holobiont evolution, population theory for the hologenome, published in the American Naturalist. Today, Joan Roughgarden very nicely agreed to talk about theoretical microbiology, holobionts, lytic-lysogenic switch, and merger versus major transition. Joan, thank you so much for agreeing to give this talk, and the floor is yours.
Well, thank you so much, Tom, and it's lovely to see you again. And I wish I were there in France in the summer. It's just a lovely prospect. You're very lucky to be there. So thank you for the opportunity to talk about my most recent work in this area. And so I'd like to talk about a collection of topics which I'm bundling under the title, the rubric of theoretical microbiology. And then specifically about holobionts, as I said, the lytic-lysogenic switch, and a critique really of the notion of major transitions and its possible replacement with the notion of a merger. Now, let's see. This worked a minute ago. Let's see if it still works. Okay. Now, since I know a lot of you folks are not all that familiar with ecology, let me just mention, a really big picture overview as to what ecology is and what its relationship is to evolutionary biology and so forth. Now, ecology basically is the skeleton of ecology is arranged according to levels of organization. So any ecology textbook, for example, that you look at would begin with at the individual level topics pertaining to animal foraging and plant physiology. Then there's the next level up, if you will, is single populations, and then interacting populations, and then ecosystem flows and ecosystems. And then ecosystem is the community, the union of the community with the physical environment. And then there's global change. So any ecology textbook will have this. And the concepts for this subject really begin with Malthus in 1800 and the notion of experimental exponential growth, and then leading in the turn of the century here, the people who added resource limitation to exponential growth to generate the logistic equation. And then there are all these other folks out here. And the key, what I want to point out right here is that this is all of these ideas are occurring in the 40s, the 20s, 40s, 60s, and so forth. And we have a whole lot of people. So the origin of ecology is it involves lots of people. There's no single figure that stands out. And and you want to contrast this with evolution, because with evolution we have Darwin in the 1850s, and then you have the formation of evolutionary biology today in the 40s, 50s, and 60s with Fisher, Wright, Haldane, so that the integration of Darwin's work with Mendel's work gave rise to population genetics and basic evolutionary theory as we know it. So that we have ecology forming as a discipline in parallel with evolutionary biology. But unlike evolutionary biology, there's no single figure, whereas in evolutionary biology, you have Darwin as the single lead figure. But as a discipline, it's developed parallel to evolution. Now, in the 70s and 80s and into the 90s, we had the introduction of evolutionary thinking into the ecological framework that all these folks originated. And that gave rise to optimal foraging theory, game theory, RNK selection. And the point is that the introduction of evolutionary thinking spanned at least three of the levels of organization in ecology. And I think we might be at a time where there's another major infusion of ideas into ecology, which is what I'm calling theoretical microbiology, because we have the notion of symbiogenesis over here, which is the origin, pertains to things like the origin of the mitochondria and the origin of the nucleus, potentially, the holobiont, which we'll have a lot more to say about, and then the Gaia hypothesis that pertains to the roles of microorganisms at a global scale. So that the impact of microorganisms spans the entire breadth of ecology, and I think we might be looking at a period where there's a major where ecology is augmented in a major way, and a lot of this is due to Lynn Margulis's work. So I hope that orients you a little bit, and that might be helpful. Now, turning specifically to holobionts and the notion of a holobiont. Now, the facts that precede the idea of a holobiont has to do with microbiome host integration and the discovery about how extensive this is. And quoting from the references down below, we have that we are colonized by microbes at birth. Nearly half of our cells are bacterial. Each of us houses about 160 major bacterial species. And Scott Gilbert has a wonderful title to an article he wrote, which is called A Symbiotic View of Life. We have never been individuals. And the whole notion of an individual is deconstructed somewhat by the realization that all these microbes are rolling around inside us. And physiologically, over 30% of the metabolites in mammalian blood are products of microbes. And these chemicals maintain and coordinate our nervous system, skeletal system, digestive systems. I mean, we just plain wouldn't work without all these products of microbes. And developmentally, guts cannot be formed without the help of microbes, because microbes activate gene expression in the cells designed to become the gut. The mammalian brain does not form without microbes. And so from corals to sponges to ticks to termites to legumes to literally every multicellular organism has all these critters inside it. Now, that's led to some definitions here. A microbiome, and so I use or take as a point of departure Josh Lederberg's definition of a microbiome, which is an ecological community of commensal, symbiotic, and pathogenic microorganisms that share the body space of a host. I love the phrase body space. And I've sort of amended this to say that an ecological community occupies a neighborhood. So it's still a spatial concept, a neighborhood controlled by a host, where there's a notion of a host controlling this neighborhood. And so it is hierarchical. And a holobiont then is a composite organism consisting of a host together with its microbiome. It's the union of the host with its microbiome, and that's due to Lynn Margulis. And again, I've amended this slightly to indicate that it's a functional unit where the host is functionally integrated with its microbiome, and leaving aside whether or not the holobiont is also a evolutionary unit, which is quite debatable, and as you'll see, not even necessarily a good way to think of the way holobionts evolve, because it's not a unit. But what is a unit is the functional unit. And a hologenome is the union of all the host genes with all the genes in its microbiome. And that's due to Jean Rosenberg and Ilana Silber-Rosenberg. And a hologenotype is when it comes to modeling the hologenome, it's useful to introduce the notion of the hologenotype, which is the configuration of the hologenome in a particular holobiont, a particular instance of it. In holobiont selection, then, is differential reproduction and our survival of holobionts depending on the hologenotypes. Now, here's a diagram of sort of holobiont evolution, if you will. And let me go take you through this. Now, we start with a bunch of holobionts, and a holobiont, so let's say one of them over here, holobionts would then have several hologenotypes, because the holobiont will have, say, the genes from two different microbes right here, a green one and a brown one. And it would also have genes in its nucleus, say a red gear and a blue gear. And so if there are two host alleles in special case, of two host alleles and two microbial strains, it would give rise to this many possible hologenotypes. And the way it works is that, to begin with, the host right here reproduces by random union of gametes or random mating, which stocks the nucleus right here with genes in a Hardy-Weinberg ratio. And there's a host gamete pool, at least abstractly, But of course, in some marine organisms, that's really out there where they release a sperm and egg actually into the water, and you actually see a host gamete pool in the water. Um, and then at the same or parallel to this, you have the microbes, which are in an environmental source pool. And so here you have the two kinds of microbes in this example. And then they colonize the juvenile hosts right here by Poisson sampling. And Poisson sampling is the sort of, just so we're on the same page here, is the kind of sampling that you experience with a Geiger counter. So so um, a host right here, could get a, a, a green microbe or a brown microbe. So it's like think of a Geiger counter and go when a radioactive particle hits, go two of them hit. So that's what's going on here, is that each one of these gets hit with a microbe, and then this one gets hit with a microbe, and this one might get hit with two microbes. But also, one of them might not be hit by any microbes, and so it would be empty. So these are the possible hologenotypes that you can have after the colonization, the microbial colonization. And then once they're colonized, then there's internal population dynamics. So the microbes grow and come to equilibrium depending on the genetic background, which is what that's supposed to indicate. And then moving over here, these now mature holobionts release gametes into the nuclear pool, and they also release algae into the microbial source pool, and the process repeats. So that's the basic schematic for holobiont evolution. Now, it's very appealing to think of the evolution within the microbial component of the hologenome as occurring completely in parallel with the nuclear part. But the devil's in the details, and it doesn't, well, conceptually it looks parallel right here. So we have the nuclear part operating in parallel to the microbial part. The nature of the colonization is different. This is binomial sampling of the gametes, giving rise to the binomial or Hardy-Weinberg ratios. But this is Poisson sampling up here. And the problem is that the Poisson sampling is not neutral to the selection on the microbes. So it imparts a bias, if you will. And that bias is a central bias. So this is a simulation and you could, in the paper I've sent you, you can read all the equations for this, but suppose you have an array of 100, this is 100 by 100 holobionts sitting out there. And then suppose the two microbes are completely neutral. So if you have two alleles that are completely neutral, then the Hardy-Weinberg frequencies don't change through time. That's the Hardy-Weinberg law, that unless there's actually a force like selection or mutation or drift or something like that, the Hardy-Weinberg ratios and the gene frequencies persist unchanged through time. But that isn't the case for neutral microbes because of the Poisson sampling. And so if you started out, for example, with a microbial source pool, which was mostly red or brown algae and only some green algae, you see that Initially, sure, most of the microbes, most of the holobionts are colonized by the red algae because they were most abundant. But eventually, you wind up with this stationary distribution of the ratio. Conversely, if you started out mostly with green, then yes, initially it's mostly green, but it still converges to the same equilibrium right here, which is 50-50. So any selection that's operating has to operate against this background of a central tendency brought about by the colonization process, together with the internal dynamics leading to an internal within holobiont equilibrium between the two parties. So that then means that selection at the holobiont level can't be as effective as selection on the nuclear level. So if you had a gene, for example, that you wanted to have fixed in a population of holobionts, you should put that gene in the nucleus and not in a microbe, because the selection can't be as effective if the gene is carried in a microbe, because the microbe dynamics lead to a, a composite outcome of the central tendency from the sampling process together with the selection itself. So what could happen instead or in addition? And so I have another simulation for you here, and this is my first attempt at making an animation. So this is really pretty amateurish. But another possibility is that the host could really control the entry. So instead of having Poisson sampling of the, the the microbial pool, source pool, the host could be active in this. And so here's our holobiont right here. And imagine that there are three types of microbes that can colonize it. There's a microbe which is actually parasitic, which is in brown here. There's one that's very mutualistic. So this would be like an algae that gives a lot of photosynthate to its host. And this one in olive is one which is not quite as altruistic. So we have a parasitic one, a mutualistic one, and a grudging mutualist. And first of all, as you'll see, so these are the contributions of the host, as indicated over here on the left-hand side. Now, and Tom, as you know, has been very active in talking about the role of antibodies and the immune system in determining individuality, and I think his work on this offers a lot of insight to this process. And so there's a host antibody response, and the host has no antibody response to the mutualistic microbe. It just loves them. It has some antibody response to the olive one, and it has a strong antibody response to the parasitic one. And in keeping with the militaristic tone of our life these days, I have indicated the antibody response as a little tank. And so let's see what happens when this process occurs. So this should be so this is going to run here for about 30 seconds. First of all, if the mutualist finds the carrying capacity within the host to be sufficiently low, it can't get in anyway. It's only if the carrying capacity of the host is sufficiently high that anything can happen. And so first of all, the low one just can't even get in. It's not enough there for it. But if it's high enough, then we get this possible colonization sequence. So the parasitic one gets in, but is immediately kicked out. So then the mutualist gets in and it is allowed to stay. And that's followed by the grudging mutualist, I guess I'll call it, who doesn't give as much to the host and benefit and uses some of it for itself. And it goes in and then it kicks out by competition, by competitive exclusion, the one that was already there, leading to this as the final configuration, where you have some antibody presence and a symbiont who's the grudging mutualist. And so this is then an integrated host, microbiome host. This is an integrated entity. It's not ideal from either party's point of view, but it's nonetheless viable. And they're better off this way than they would be by themselves. So this I'm calling the adaptive holobiont assembly via host orchestrated species sorting, which is a mouthful to say. But species sorting is a phrase used in microbiology a lot. And so microbiologists say that, the bacteria are everywhere and it's the environment that selects which ones can live in any one spot. And this is just an extension of that idea where the species sorting is orchestrated by the host. So this yields host microbiome integration. And so this is an alternative process to thinking of holobiont selection as being the cause of host microbiome integration. And I think with respect to holobionts, the main question to ask is what can explain the functional integration? I mean, that seems to be the fundamental evolutionary question to address. And so this is one possible answer. And as I say, it could possibly be due to holobiont selection, but it just seems a little less plausible because of the limitations on the effectiveness of holobiont selection. However, you could put them both together. They're not mutually exclusive, so that you could augment the diagram I gave you before, but instead of having Poisson sampling here, you could have host species sorting, where the host determines whether like a green one can come in, but a brown one can't, and so forth. And so far, I haven't, no one's modeled the combination of these two processes, which would lead presumably to lots of neat mathematical outcomes and so forth. But this is the basic idea that you still could have species or holobiont selection, but merely that the initial genotypes hologenotypes each generation were set by species sorting. Of course, it's understood in here that there's selection in the nucleus on the capability of the host to do the sorting. So moving right along, let's talk about viruses. Now, as I started to work, first of all, I started working on viruses because the overall objective of the project would be to look at a so-called tripartite holobiont, which is then a virus and a bacteria and a host. And I thought that would be a pretty simple task, and then I would just shop around for a good virus model and then weld it to the holobiont model I just showed you, and then we'd have a tripartite holobiont system. However, it turns out that there's that there's not really a satisfactory, or at least in my view, wasn't a satisfactory model for viral phage for virus bacteria population dynamics. And so I've had to get involved for the last year and a half, really, on what began as a digression into developing a model for phage bacteria population dynamics. And along the way, I discovered a lot of things about viruses I didn't know, which are kind of neat. And one of them is just how many there are. So the number of prokaryotes on Earth is on the order of 10 to the 30. And the number of virus is 10 times the number of prokaryotes. So that makes the number of virus on the Earth, the number is on the order of 10 to the 31. So now what does that mean? 10 to the 31, how do you wrap your mind around that? Well, you may recall that Carl Sagan, I don't know if you recall this, but Carl Sagan used to say that there are more stars in the universe than there are grains of sand. And so an astrophysicist decided to check it out. And that's this guy, Plate, right here, who published this in the Scientific American. So it's sort of like a popular article. But he nonetheless did calculate that the number of grains of sand on all the Earth's beaches is only 10 to the 20. And the number of stars is a meager 10 to the 19th. So there are actually more grains of sand than there are stars in the universe. But of course, all of this is 10 orders of magnitude lower than the number of viruses. And so, the potential of viruses. Now, I mean, viruses are organisms. There's no point in talking about them as arguing whether they're alive or not. They're evolutionary agents. They have genes in them and they evolve. And so the smart thing to do is to just treat them as organisms. They might not have their own metabolism, but that doesn't matter. They have their own genomes. Now, the basic question in viruses is this transition between the lytic and the lysogenic stage. And the jargon, there's a lot of jargon in this field. A virulent virus is one that can't switch. It's just always a parasitic on the host cell. And a temperate one has the capability of switching between being lytic and being lysogenic. And lytic means that the virus undergoes this life cycle right here, where there's a bacterial pool. Again, we have Poisson sampling for a virion pool. So a virion is just a free virus particle. There's a pool of these out in the environment, Poisson sampling. And so again, we get some bacteria are infected and some aren't. And the infected ones then go and grow within the bacterial cell and lyse, which means they break out of the cell wall and then repopulate the virion pool, whereas the ones that are not infected, the bacteria just reproduce by themselves. So that's the lytic cycle, and that's the parasitic cycle. Now, the lysogenic cycle is really neat. It's where where the virus, which is indicated here by the helix, the little colored helix, they just simply insert into the bacterial pool, in the bacterial chromosome. So they insert themselves in the bacterial cell and just simply reproduce with exactly the bacterial growth rate. So they merge, and a point we'll come to later. So we get this merger of the bacteria of the virus with the bacteria. So a temperate bacterium then has a choice. A temperate virus has a choice of whether to reproduce this way or to reproduce this way. So if you model this process, you come up with a pretty nice criterion for when to do one or the other. So this is, as you see in the manuscript, you have On the left-hand side right here, we have the so-called burst size of the virus. So the left side here refers to the lytic, and the right-hand side refers to the lysogenic. So, so the reproduction that a virus can have, if it's going to be lytic, is that it's going to have a burst size. So it's going to it's going to lyse that many particles. And then A over here is the absorption, because once they're released into the viral pool, they then have to then colonize another virus, another bacteria for the cycle to repeat. So the product of the absorption probability times the burst size has got to be greater than R. And R over here is the growth rate just of the bacteria. So if it can grow by being nasty more than it can grow by being kind or by being integrated, then it should go ahead and light and be lytic. On the other hand, if its production when it's lytic is less than its production would be by just simply incorporating into the virus, it should go ahead and be lysogenic. So my model for the conditions for the switch is just this simple criterion right here, which comes out of the differential equations for the population dynamics of this system. So downstream sometime, if I the goal eventually of looking at the tripartite holobiont is in the distant future, and I might be able to merge this together with the holobiont model previously. Now let me turn to an interesting issue, though, that comes up, and I'm going to quote this table right here from the philosopher Matthew Herron, and I encountered this in Biology and Philosophy, a journal that I'm sure many of you know. It's an interesting review article of the notion of the major transitions. And notion of major transitions, as I'm sure you're all aware, have been with us since 1995, when Maynard Smith and Zameri talked about major events in evolution, or they call them major transitions, which is the origin, things like the origin of multicellularity and the origin of eukaryotes, is going from a prokaryote to a eukaryote. And this review article points out, though, that that today, looked at today, the clarity of these transitions is not terribly obvious. Very different researchers have different criteria. They allow for different kinds of things as phenomena as being major transitions, and it casts doubt, in my view, on the utility of the major transition idea in today's in retrospect. And moreover, it doesn't address something like a holobiont, because no one's claiming that a holobiont is a major transition, yet nonetheless it is a functional entity. And so I've been suggesting, or have suggested, that there's the notion of a merger, which would be maybe a replacement for the notion of a transition and individuality. And transitions, as I just mentioned, are directional. They assume vertical transmission, and they have disputed criteria. Now, the notion of a merger, so there's an area of economic theory called industrial organization, and it's all about firms and how firms can merge, and then how they can disassociate. Part of a firm could be carved out, and divisions could be released. And another interesting idea in this industrial organization is called the principal agent problem. And that's basically about a firm in which there is again, these are hierarchical. So there'll be an owner of the firm or the so-called principal agent, the one who controls it. And the task of the principal agent is to develop a compensation structure or an incentive, a system of compensation for the workers, which aligns the workers' incentives with the incentive with the production of the firm. And so the principal agent problem involves how the controller, the owner, the principal agent manages the 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 workers' compensation to bring the workers' incentive in alignment. And then you get an efficient firm and everyone's better off. And if we think about the idea of game theory, which has had a lot of impact in behavioral ecology and understanding, and game theory is just so that everyone's clear about this. So game theory is basically optimization theory for two or more interacting parties. So it's just an extension of optimization theory, but to two or more parties. And it had a big impact there. And it seems to me like we could mine, if I could use that word, mine or explore the ideas in the theory of industrial organization to understand things like mergers and spin-offs and so on. And that this gets to, I think, what the major transitions are. They are mergers which then lead to spin-offs. Whereas something like a holobiont is a looser kind of merger, and it allows for the parties, but it allows for the parties to go their own separate ways, and it's still the job of the host within a holobiont to potentially align the incentives of the microbiome within it. Anyway, I think that's an idea that a tack that might be worth exploring. It's non-directional, it allows horizontal transmission, and the criteria are loose. So I'm just throwing that out there. And I think it might be worth exploring. So for a theoretical microbiology, it seems to me like these are features that would distinguish it from previous theoretical constructs. So first of all, environmental source pools are fundamental to it. So that it's not just population dynamics where the population is self-contained. All of these things are populations that are partially in the environment. And so the environment is a first-class element or object in the modeling. And secondly, vertical transmission is not privileged. And much of evolutionary thinking, does privilege vertical transmission. And Lewontin's stuff on the conditions for natural selection also privileges vertical transmission. And I just think that's not useful anymore. And then there's the notion of adaptive colonization. And then I think also theoretical microbiology has to include viruses. It's not just about bacteria. And as you know, the people studying viruses are a different peer group from the people studying bacteria by and large. And I think we need to make sure that that disciplinary boundary is dissolved. And then lastly, as I was just discussing, we might explore the idea of mergers instead of transitions. So there we are, Tom, and thanks so much for the invitation. And this is of interest and maybe stimulating.
Very much so. So thank you very much. That was extremely clear and very, very interesting. So um, if this is okay for you, maybe you could stop sharing so that we could see everyone on our respective screens. Yeah,
Yeah. Oh, there
okay.
you go. Okay.
Excellent. Thank you very much. So one thing I would like to ask everyone is to very quickly, before asking your question or making your comment, maybe say in one sentence who you are, because depending on whether you are uh, an expert in viruses or bacteria or a philosopher, it might make a difference. So let me see if I can see the gallery. Um, I don't see any questions for now, so I have many questions. So I can start with a simple question, which would be around the fact that um, it seems to me that you have developed a functional first approach to holobionts in the sense that you insist on the importance of functional unity and physiological integration from this point of view. And then you draw some consequences, including in terms of evolution of this approach to the holobiont. And I very much like that, but I had two questions about this. One is that some people say that when you have cases like insects or some arthropods, when you have, in fact, a very reliable vertical transmission of the microbiome to the next generation, in that case, we could say that the holobiont is an evolutionary unit. So I just wanted to know if you agree on that. And the second question, which is more conceptual, is that I've seen some people over the last 10 years or so who have said that the holobiont literature has been too host-centric. And in a way, I was asking myself, would you agree that your approach, in a way, is host-centric or not? And what would be the consequences of that? So two different questions here.
Um, so with respect to insects where there is vertical transmission of the microbiome, um, that could happen. And of course, in my I'm agnostic on it. I mean, if it is, it is. And um, I don't take that as the generic case. However, it seems like, um, and for the modeling that I've done, for example, I could always add vertical transmission in addition to horizontal, or have some ratio of vertical versus horizontal and so on. But it seems like the conceptual advance comes from looking at it as horizontal, because that then leads to the notion that the environmental source pool is the logical counterpart of the gamete pool, and that's a nice idea. Whereas trying to shoehorn the microbes as though they were being vertically transmitted, uh, it is not a step forward. I mean, it might be true, but it's not a conceptual step forward. Furthermore, it's not clear that that's going to work very well, because the vertical transmission, even in that case, would not necessarily be Mendelian. And so when you get into the nitty-gritty of the population genetic mechanics of it, just vertical transmission isn't enough. It has to be Mendelian in order to give you neutral, in order for the transmission process to be selectively neutral. And if the transmission process is not selectively neutral, then you have the same kind of problem that I've already pointed out with the horizontal. So yeah, yeah, no one would deny the vertical transmission that does take place in insects, but its conceptual significance, it seems to me, to be more arguable. Now, as to whether or not the holobiont concept is host-centric, yes, it is, and it should be, in my view. host-centric because it's about the host controlling a microbiome. Now, otherwise, you could just have a microbiome host interaction, like a binary interaction. But the whole spirit of the holobiont is that it's a it's a microbiome within a host or in a host's zone, and therefore um, I don't regard it as a criticism that it's host-centric. I think that's a feature and an important one. And for cases which aren't subject to host control, then we're just dealing with a binary interaction between a host and a microbe. And that's nothing particularly novel about that. It's just another binary interaction, like a predator-prey or a host-parasite or a competition binary interaction. But the whole notion of the holobiont is that you have this integrated package, and it's in something. So that's the definition of the holobiont in my view. And if someone wants to study some other kind of system, then fine. But yeah, that's what I'm modeling. That's what the system is.
Yeah, okay, very good. Thank you very much. Any questions in the audience, or maybe comments or things that were not clear, which I doubt, given that the talk was incredibly clear and well structured. Any questions? Um, if you want to ask a question, you can just raise your hand in Zoom, or maybe just turn your camera on and ask a question. I'm surprised we don't have more questions. I have more questions, but it would be a pity if I was the only one asking questions. So a quick question. I hope people will have questions. I had another question about um, the distinction between fraternal and egalitarian transitions, and I was thinking that there was this distinction made by David Queller about egalitarian transitions and fraternal transitions. And it was not entirely clear to me to what extent the merger or mergers you described was could be described as part of an egalitarian transition in evolution.
Oh, well, I'm not familiar with that, but it sounds like it could, yes. Uh, yeah.
Okay, we could talk later about this, but I think it could
Yeah.
be because I think your model is different, so it would be interesting to see to what extent the theory behind that is going to be different from the one that Quater has suggested.
Okay.
Okay, no, no, okay. I Let's see if some people, I'm also checking in the chat. So, all right. Um, um, a small thing I would like to discuss with you later, maybe, John, is the question of there is now very good evidence that bacteria have very complex immune systems. So I was thinking, of course, it would not be antibodies, but you could still incorporate your views about immunity and the nice animation, even for the very interesting model about bacteria and viruses. So that, yeah, the tripartite model could also incorporate the way in which, of course, bacteria can be selective on which viruses they allow to get in a way.
Yeah, that's interesting. No, I didn't know that.
Yeah, yeah, I can share with you
But
some papers, but
you're the expert on this. I mean, you have found immune-like systems everywhere, and when we first met years ago, I didn't realize that.
I haven't found those systems, unfortunately, but it's good to follow this and to realize that
Yeah,
indeed, in the end, I think there's a quasi consensus at the moment that immune systems exist in every living thing. So it's interesting
yeah,
also because in terms
yeah,
of modeling, it might have consequences about adding the components that you mentioned, basically in every living thing. So yeah, so that just extends what you said. So this is why I was thinking about that.
yeah. That's remarkable, really.
So there's now a question in the chat or comment, I don't know, by Emily saying, so would you say that the holobiont counts as an individual in the immunological definition of individuality because of the host-mediated species sorting? What do you think, John? It's a question about
Yes,
immunology.
I think a holobiont does count as an individual in the same sense that a firm does. That like what is does it's the notion of a corporate, like it's the notion of a corporation, and so a corporation has divisions and so forth, and but to the extent that but a corporation would be taxed as an entity and has rights associated with it. So I think that a holobiont is an individual. It's a functional individual, and I have no problem with that. It's just that the host isn't the individual; it's the host and the microbiome which constitutes the individual.
This is also related to what you were saying with the paper by Scott and others. We have never been individuals. I think indeed the conclusion is rather that hosts, as we have known them, are not individuals, but we can still maintain, I think, a notion of individuality. As you just said, we have to expand or change it a little bit, but not necessarily conclude that there's no individuals anymore in nature.
Right, right, exactly. Yeah.
Okay, so if there are no other questions, I really want to thank you once again, John. That was extremely good, clear, and interesting. So thank you very much, and I hope that it was interesting for everyone, and that, John, we can continue our discussions in the future.
Right, great. So thank you all.
Thank you very, very much and see you soon. Bye, everyone. Bye.
Bye.